Hello, everyone. I think we're still letting a few families join in. We'll give it maybe another 10 seconds or so.
So I just quickly have to take something from my foster dog. I'm going to be right back on screen here, 1 second.
Sure. All right, I think we'll get started. Hello, everyone, and welcome to the Families RX Hope Table Talk series. Today's session is our 9th talk, and we will be learning about something that all of us are very excited to hear about, the progress in discovering new drugs for SCN2A. This session is being recorded. My name is Sean Egan, and I'm on the Families SCN2A Foundation board. We are very excited to be bringing this virtual education experience to our global community. During these sessions, the families and caregivers of patients with SCN2A related disorders will be referred to as the experts, and the presenter will be the professional. The goal of our virtual table talk is to allow experts unprecedented access to professional researchers, clinicians working diligently to find a cure for our children. This is meant to be a casual environment, and your questions and comments are encouraged, as we are all here to learn from one another. To keep things running smoothly, please keep your audio on mute and use the reactions buttons to raise your hand if you would like to talk. Alternatively, you can send questions through the chat. If you haven't already, please introduce yourself and your SCN2A child in the chat. Maybe perhaps their name, their age, and how SCN2A presents. My daughter Harper is 4. She presents with seizures, chorea, and hypotonia. You could also give where you live, so we can kind of see where we all live relative to one another. I now have the real pleasure to hand this over to Dr. Kathrin Meyer from Nationwide Children's in Columbus, Ohio. This is an institute and research center that has been a real focal point for developing cutting-edge transformational new medicines for pediatric neurological conditions. Kathrin?
All right, thank you, Sean. I really appreciate it. Let me share my screen. I have a little introduction about myself in the presentation. Can you see this? All right, okay. And should I get started, or are we still waiting on people to join? Okay, I'll start. Just make sure I see my slides as well, moving this around. Okay, so I'm Kathrin Meyer. I'm originally from Switzerland, but moved to the United States almost 11 years ago to work with Brian Casper at the Center for Gene Therapy to develop gene therapies for neurological and neurodegenerative diseases. These are my disclosures that I have to bring up real quick. And I wanted to introduce the Center for Gene Therapy a little bit to you guys. It was established originally by Drs. Phil Johnson and Dr. Jerry Mendel. Our current director is Dr. Flanagan since 2017, I believe. We focus on neuromuscular disorders of children and adults, and we have translated 13 AAV gene therapies to the clinic in the last 10 years. So really is a very strong pace. And there's also a good group of successful gene therapy startup companies that were founded by researchers of this institute, including Avexis, Myonexus, Celenex, Milo, and Abiona. My lab, here you see a picture. It's a little bit older. I need to get a new picture soon. We focus on neurodegenerative and neurological disorders. And I personally have been involved in 4 successful IND submissions, so investigational new drug applications to the FDA. Those were all gene therapies for spinal muscular atrophy, both intravenous delivery, intrathecal delivery, and then 2 Batten disease programs. Batten disease are seizure disorders that are caused by lysosomal storage problems. And the next IND submission I'm very excited about should be happening probably within the next days, but just to be careful saying at least within a month. So we should have a 5th submission, if successful, a clinical trial to be starting still this year as well. And of course, my lab focuses on gene therapy. I'm at the Center for Gene Therapy in Columbus, Ohio, but we also do a lot of in vitro modeling. And I hope you'll see today why I think this is very important. Real quick overview over gene therapy. I don't know if everybody is familiar with it, but it really just means the transfer of genetic material to treat or to prevent diseases. And there are different types of gene therapies, and the most straightforward ones are gene replacements. We call it gene replacement, even though it really just means to bring a healthy copy of a non-functional gene or a reduced functional gene into the cells. So we're not really replacing anything, we're adding an additional copy. The second option is to gene silencing with different kinds of tools. We can reduce expression of an aberrant toxic RNA or protein. And then the third option is to actually do gene correction. So really go into the genetic code of the patient and make that change back to normal, basically. Oftentimes this is done in tissue culture. So for blood cells, blood diseases, for example, you can isolate the cells, can fix them, and then put them back into the patient. That is, the current applications are mainly focusing on the ex vivo change of cells. All right, so how is that important for SCN2A? And some of you might have seen some of these slides. I just want to remind you real quick. SCN2A, unfortunately, is not a good fit for a gene replacement strategy because the gene is too big. We can't fit it in the AAV vectors. So there are potential approaches where you can split up the gene in multiple sections and then use several vectors to bring it into the cells. But there are risks to it because you can't guarantee that every cell will get both vectors. And so then you're going to make partial proteins that can be toxic as well. Gene silencing might be an application for patients with gain-of-function mutations, but it will still not restore the proper function. So most likely not the best option for SCN2 either. For gene correction in the nervous system, you cannot isolate the cells and put them back in, so that is problematic, but there are technologies that are currently developed that might be feasible for SCN2A. The one you probably heard about the most is CRISPR-Cas9. It's a system that has been discovered in bacteria, a protein that can go into the cell and really cleave the genetic material and make changes. The problem is that this approach is mutation specific in many cases. So you would have to develop a new approach for every single mutation or for many different pieces of the gene. Also, there are off-target effects. So this protein can cleave somewhere else, and that can cause mutations, and it could be potentially harmful as well. And then there are approaches that use modified versions of these proteins, but they have to be permanently expressed. And that is also a little bit concerning because these are bacterial proteins. And if you express a bacterial protein permanently in the nervous system, this could cause inflammation as well. So that's something that I'm personally a little bit worried about. The good part about this strategy is you don't actually need to understand what the consequence of the mutation is. And in many cases, we don't know exactly, is it a gain of function, is it a loss of function, or a mix? In this case, it doesn't matter because you're just changing the problem. Another variant of fixing the gene is actually not working with the gene itself, but with the working copy, which we call mRNA. So that's called splicing correction or trans-splicing. And what is nice is you would change a piece of the entire RNA, and you can address both gain and loss of function in that area of the protein. Just to quickly explain that a little bit, how that works. So you have a gene that's encoded in your genome. From that gene, you will make that messenger copy, that working copy, by removing some crucial pieces in between that just have additional information that doesn't go into the protein. And I can just represent that here with this sentence, a big dog ran and sat. So each of these squares or triangles is encoding one of the pieces that goes into the working copy. And from the working copy is actually what we make protein from. Now, if you have a mutation that leads to a stop codon in the middle here, then ultimately what happens is you don't know anything about the rest of the information. If you have a frame shift, that means that the entire second part after that mutation is going to be completely messed up. So you'd still be able to read a big dog, but the second part, you're not going to be able to understand. And that oftentimes leads to proteins with toxic gain of function, or they are rapidly degraded, and so you don't make any protein. Missense mutations would just basically lead to a tiny change in one letter, like you see here. So what can be done with transsplicing is that you take a piece that you know contains mutations, and you basically tell the cellular machinery to include that piece instead of the piece that contains the mutation in your working copy. So this would affect the working copy. It wouldn't change the genetic code of the patient. And you'd be able to make a full-length normal protein. And as I said, the beauty of that is it doesn't matter what type of mutation you have, as long as it's in this section here. So these are interesting approaches. The main issue with transsplicing is though that it is very inefficient. So we need to find better ways to tell the cellular machinery to include that new piece that we're bringing into the cell. And that's something that we're working on at Nationwide. The other way to do this is to modulate just production levels. And there are multiple ways to do that. There are small molecules that can increase expression. There are special CRISPR approaches, and then also new technologies that we are working on at Nationwide. So just real quick, as you know, in SCN2A, the patients have one healthy copy of the chromosome in most cases that still produces protein, and then the second chromosome that doesn't produce protein or produces the mutated protein. So what can be done is basically ignore the second chromosome and just focus on the first chromosome and try to upregulate the expression of the healthy copy. And this can be done, as I said, with small molecules, but we at Nationwide are working on a gene therapy approach that doesn't require the delivery of any bacterial proteins to be able to do this as well. All right. So the main focuses of my lab are to make the transsplicing more efficient. We have multiple transsplicing constructs that we're working with, but to date, they are not efficient enough to move forward with a preclinical IND enabling study. And then the second technology is very new. So we're upregulating the promoter using non-bacterial factors. All right, so just quick summary on the gene therapy side. Gene therapy for SCN2A is very challenging for many reasons because of the complex function, but also the size of the protein. But there are multiple groups that are working on solutions, not only us. And I think there are some promising approaches that are moving forward, but it's going to require more research. So I think this is going to still take a little bit of time until this will be ready for clinic. DNA or RNA could be a target for gene therapy, and most likely it's not going to be a fit for all situation, as the disease pattern and the type of mutations are just too complex in SCN2A. So that brings me to the next question: how do we determine which patients can benefit from a therapeutic approach? And in my opinion, the best way to do that is cellular disease modeling, so you can include as many different mutations as you can. Real quick, how do we do this? We can get patient samples, for example, from skin or blood, which you can see down here, and we can reprogram those into different types of cells. And what you probably heard most often is reprogramming into induced pluripotent stem cells, which you see those 4 blobs over here, which are what we call stem cell colonies. We use embryonic factors to basically tell the cells to go back to their embryonic stage. The good part about this technology is you can make almost any cell type in the body from these stem cells. The bad part is that you're working with what we call a clone. So one of these stem cell colonies originated from a single cell and it was then amplified. But not all the cells in your body are the same. And sometimes you get bias that is introduced by this technology just because that one cell that you made the colony from had a problem to start with that wasn't relevant actually to the disease. The second problem is that you erase a lot of the memory of the cell that it acquired during its lifetime living with the disease. So you get back to a milder disease phenotype in some cases. There are other ways to reprogram cells, and the one that I like most is to use pretty much the same system, but push the cells right away into neuronal progenitor cells. That allows us to move faster. There is no clonal selection. We're working with a pool of cells, and the maintenance is much less time consuming, so we can do more experiments, but we can still make different cell types of the nervous system from these progenitor cells as well. So here's an example of how these cells look. On the left side, you have skin fibroblasts, and then within about 2 weeks, 2 to 3 weeks, depending on the cell line, we can push them into these much smaller cells that we call neural progenitor cells, and they tend to aggregate and stick together as well. So with this method, we can model patients that have sporadic diseases, which we don't know what the origin is, or patient mutations that we don't have a mouse model for example. There is another technology that we recently started using, which is even faster, and it's a chemical conversion. So we use the skin cells and we use different chemicals to just push them directly into neurons. And it only takes about 7 days. And here you can see some pictures of a healthy control cell line compared to 3 different patient cell lines suffering from SCN2A mutations. And what we found is that at least in some of the mutations, we do see a reduced number of neurons after 7 days. So there's something wrong either with the differentiation or with the survival of these cells. And that's something we're studying at the moment. We also have, and I'm going to talk about this a little bit more, but there is a small molecule that we've been working on that can increase the survival of these neurons at 7-8 days post-differentiation as well. Another thing that I kind of want to point out, and that's last time in my talk that I talked about neurons, is that in many neurological diseases that I work in, the main focus is always on the neuron, because the neuron shows the strongest disease phenotype, or we're just more used to see the neuronal phenotype, and they are oftentimes the first cells to die. However, the number of neurons is actually smaller in the brain compared to other cell types. Astrocytes, for example, are extremely important support cells. They have a lot of metabolic functions. There's also microglia, which is the immune cell of the nervous system that has an impact on seizures as well. And there are oligodendrocytes that could be impacted too. And people are oftentimes not paying attention to these additional cell types. And they really impact neuronal health and disease progression in many diseases. And they might be good targets for therapeutic therapies as well. So what we do is we use this reprogramming method to make neural progenitors. And then I really like to work with the astrocytes because of their important function, but also because we can run the assays much more standardized. They're very easy to work with. Here is one of the assays that we're using. So we're taking neural progenitor cells from patients, we make astrocytes, and then we co-culture them with green fluorescent neurons from mice. We use them from mice because they are much faster to generate and we can make them in large numbers. So when you put them together, we can use automated plate readers to really understand how the human cells impact the health of the neurons. And the green fluorescent neurons are here just depicted in black. But what you can see is that in a healthy control, they do clump together a little bit, but they have those nice long extensions, which are the neurites. And then the SCN2A cell lines show a very severe phenotype with either reduced survival or shorter neurites as well. So this is just a quantification of what I just showed you, that the reduction of the survival of the mouse neurons that are in culture with the human cells. And we have this automated plate reader that we can use to now test different conditions in this co-culture system and see, you know, how, for example, small molecules or other therapies could impact the survival. Another thing that we did is we looked at the sodium uptake and the calcium uptake of the SCN2A cell lines. And, you know, it's something that I always like to do because sometimes the genetic mutation doesn't actually predict what happens in the cells because there are compensatory mechanisms that can take place. And one of the mutations that was really interesting to us is this one. This is supposed to be a loss of function that leads to an early truncation in the SCN2A gene. And this cell line should actually make 50% less SCN2A protein. However, when we looked at the sodium uptake, it was actually increased compared to healthy controls. And when we did staining and RNA analysis for SCN2A protein and RNA, we actually saw that there was more protein made compared to healthy controls. So it's always important to double check what the genetic code is predicting because of those compensatory mechanisms. And then same with the calcium signaling, there was one of the cell lines that showed an increased calcium signaling compared to the others. Based on the, this balance in the ion uptake, one of the things we wanted to look at very quickly was the mitochondria, which are important for the energy household. And thanks to families of SCN2A, we have a seahorse analyzer in the lab. So this machine here, which is called seahorse, can measure metabolics and energy household in cells. And the cells go in here, as you can see. What we found is that the SCN2A cell lines are actually producing too much energy. So they have increased energy production, and that can be very unhealthy. And this goes along with aberrant networks of the mitochondria. So either fragmentation or extreme elongation and network of the mitochondria. So there's different types of phenotypes that we're currently evaluating. What's interesting is that this elevated energy state is something that we've seen in other seizure disorders and in other neurodegenerative disorders as well. And so that helps us to look for drugs that we could potentially repurpose for SCN2A. And one of the molecules that we've been working on is copper ATSM. It's a small molecule that contains copper, and it's in clinical trials for ALS with quite promising results in phase 1. And the reason why we were excited about it is that we've seen in ALS patient cell lines a very similar energy profile. So we wanted to compare the two conditions. As you can see here, when we treat the cells with copper ATSM, we saw a reduction in the energy level. We also confirmed that with brain organoids in a collaboration with Dr. Mark Hester at Nationwide Children's Hospital. So here you basically use the stem cells, but then you mature them for we went up to 150 days, and then we were treating them with copper ATSM starting from day 90. So we did see a very similar observation in the brain organoids as well. Ultimately, in a collaboration with Dr. Kearney, we then started testing whether the in vitro model, so the disease model in a dish, would actually correlate to what we see in the mice. And we tested, this was a very small pilot study on the Q54 SCN2A mouse, which is a very severe gain of function mouse. And as you can see, compared to the untreated animals, we had a very strong effect on survival. So this was very exciting. Based on this data, we had a conversation with the company that is in clinical trials for ALS and with the FDA, and they wanted to see safety data in juvenile pigs before we could move forward with a potential clinical trial, as the drug is currently only used in adults. So that's what we've been planning, and we were hoping to start the safety study early this summer, but unfortunately we had several delays. Some of them were COVID related, some of them were not COVID related, and we still weren't able to start the study. However, we are very close now, and we're hoping that we can initiate it in Q4 of 2021. Another thing that we still need to do, and we're getting started with this, is a dose response testing in the SCN2A mouse model, because we want to make sure that the dosing paradigm is optimal for SCN2A. Just because the drug works in two different diseases doesn't mean that the dosing is optimal the same way in both cases. So that's something that we're starting, and we're going to test, what happens if you give the drug twice a day, once a day, different doses, and see how we can optimize that as well. But in addition to that, one thing that we're really interested in is to understand the mechanism of action, because the mechanism of action that we found is not what the company thought is happening. So there's probably multiple mechanisms of this drug. And the question is, if we can find the underlying mechanism that copper ATSM is targeting, maybe we can make a gene therapy out of that. And that could be really interesting for multiple diseases as well. So we have received, and thanks everybody for participating, multiple additional cell lines that we have reprogrammed, and we're now expanding the testing to see how many patient cell lines can respond to copper ATSM, so we can understand whether there are non-responders that potentially wouldn't benefit from this treatment. And then we're also testing additional drugs in our cell culture. So the drugs that we're focusing on, we have a more targeted approach. We're not doing large-scale screening, although we do have the machine to do it. I just don't have the manpower to do like large drug screens. But what we're focusing on is basically neuroprotective drugs or drugs that reduce stress of energy production, because we already have identified that as a problem, or drugs that we have heard from families and other patients that are promising for other disorders with similar pathology. So those are the kind of things that we are prioritizing in the lab. Yeah, I think that's all I wanted to say. I didn't want to take up too much time, but just wanted to thank families of SCN2A for their support. It's been, really great working with you all so far, and looking forward to continue our research.
Fantastic, Kathrin. Lots of really interesting things that you brought up kind of during your talk, from gene therapy to new novel drugs to mitochondrial conditions, which is something I've always kind of speculated when I look at my daughter. She kind of jumps out at me as somebody with mitochondrial condition, but every time I bring it up to our neurologist, they always indicate, there's no evidence to suggest such. So really fascinating and interesting. Maybe some questions that would be kind of helpful for everybody is, a lot of us on here have heard of gene therapies and kind of know roughly what they are, but maybe could you describe the differences between gene therapy and kind of other small molecule drugs that we're kind of familiar with? Maybe like some key talking points would be, their therapeutic potential, the ability to kind of treat more than once, this one and done. Is there, will they have the option to, get multiple gene therapies in the future? Or like, what are the considerations there? Yeah. And also kind of like timelines to approval. Like when we hear a small molecule drug that sounds exciting from a preclinical study compared to a gene therapy preclinically, which one of those are going to get to our kids first?
Mm-hmm. Yeah. So those are really good questions. Important for gene therapy is also to keep in mind there's multiple types of gene therapy. So there is the viral vector gene therapy, which is what I work on mostly. And then there is what we call the non-viral gene therapies, which are antisense oligonucleotides or RNA-based therapies. And they're also considered gene therapy. And I think that's sometimes confusing when you hear this. Now, the vector-derived gene therapies that we work on, they have two big advantages. The first one is that viruses have spent thousands of years figuring out how to get to the nervous system. So we're hitchhiking a system that has been optimized for thousands of years. And the spread and the distribution is just to date much better compared to the non-viral gene therapies. And the second aspect is that the viral gene therapy is permanent, or at least long lasting. So it will be a single treatment. You get a drug once, and then you don't have to go back and be re-ejected all the time, because the viruses we use, they will deposit that additional gene in the cells, and it will remain kind of almost like an additional chromosome. And that's very convenient. The drawback also is, if it is something that is harmful, then we can't take it out. That is the advantage of a small molecule or non-viral gene therapy. If it doesn't work for your kid, you can stop it. And so that's why the safety requirements are higher with viral gene therapies. The FDA wants to see usually longer safety studies. And then there are some requirements also as of what type of patients you treat first. Oftentimes, you have to start treating patients that are a little bit more severely affected, just in case there is a severe side effect that we didn't anticipate. We used to be able to move viral gene therapy relatively quickly because we do have a lot of experience. So as long as you don't change the virus, we were able to move gene therapies into the clinic, let's say within a year. Now, within a year from when you have a promising candidate. So the longest part, I think, for the gene therapy is always to find, that target in your case, because you can't fit the entire protein into the vector. So once you have a promising candidate, a promising strategy that works, then you just have to do the safety studies and some dose response, and you can get into the clinic. However, as the gene therapy field is maturing, we see that the FDA is asking for more and more additional studies. So, and also there is additional, scrutiny from the hospital, and the administration is getting bigger. So I would say now it takes about 2 years to get into the clinic from the moment you have a promising candidate, 1.5 to 2 years. There are some ways you can cut it down by running safety and dosing studies in parallel. Then it's just basically a financial risk. We would run exactly the same studies, but normally we wait until we know, yes, this is really working before we start like the big safety studies. But you can do that in parallel and you can gain some time, of course. Small molecules are faster especially if they're already FDA approved for something else. Let's say in the case of corporate DSM, the problem is the clinical trial for ALS is run in Australia. So it is not an American study, and it's in adults. If that was a drug that was in the United States and already approved for kids, you could move very, very quickly. Does that answer your question, or? Yeah, I think you're on mute, Sean. So. I can add maybe for ASOs and RNA-based gene therapies, I would also say, an advantage there is that the production is much cheaper. And, also you can move faster because it's not permanent. Gene therapy, one of the big limitations in gene therapy that is sponsored by patient foundations is the price also. Even just to make a small amount of virus for, 5 to 10 patients is about $1.5 million for a phase 1-2 clinical trial.
Yeah, thank you, Kathrin. Sorry, I was moving you to a different screen and you got lost between screens, but I think I got you
Don't
back.
worry. Yes.
Yeah, it brought up some awesome points in there. We actually have a quick question in. Do you think it's worth testing phenobutyrate in SCN2A models to see if it has a similar benefit to what STXBB1 and SLC6A1 are seeing? Is it a repurposed drug, so it may be faster to get into trials?
Yeah, definitely. We actually do have the drug in the lab, and we're definitely planning to test it. The energy profile of SLC6A1 is different from the cell lines that we have for SCN2A, at least for now. But, this is only one of the aspects of the disease, so that might not tell us everything about it. So I think it's worth testing for sure.
In your previous answer, you brought up one thing that I think is really interesting about AAV-based gene therapies is that you have the gene putting in, and then you have the AAV, and those have, different profiles and different questions. And for the AAV perspective, you have your tissue distribution and a safety profile. And once that's kind of ironed out with a dosing, like let's say there's another, like I think about SMA, which has an approved drug, Zolgensma, that's been, very safe and completely transformative. I think it uses AAV9. Once you have that profile ironed out in that dosing, like how easy is that to switch in and out genes, and how similar should the safety profile and like tissue distribution profiles be?
Yeah. So the tissue, as long as you stick to the same serotype, there's already a good amount of things that you can learn from the previous protocols or for the previous programs. So first of all, the tissue distribution will be the same, because it depends on the surface of the virus. The expression, so which cells are going to make your protein, you can steer a little bit by, the type of promoter you use. So how are you going to, it's going to be part of your construct, basically. The most important part, in my opinion, for a good efficacy is to get to as many cells as possible, especially if it's a gene that is pretty widely expressed. And that's something that we can learn from the already ongoing clinical trials. So if there's efficacy, clearly it had to be able to get to the places where it needs to go. So for SMA, it needs to go to the spinal cord, but there are also motor neurons that are in the brain. So we know we're reaching those too. And then for Batten disease, it's a seizure disorder. So that needs to go throughout the entire brain. And, it's still early in those trials, but I would say the data that has been publicly released by Amicus Therapeutic is pretty promising. So we know we're getting to the brain. The one thing that is different between the diseases is what's the tolerance level of the protein, for example. So let's say if it's a classic gene replacement, there are certain proteins that are great to have in a certain amount, but if you have too much, then it gets toxic. So that tolerance level is something that we need to test for each of the gene therapies. But yes, in terms of finding the dose and also then safety concerns in terms of the immune system, we can leverage the experience that we have from the clinic, and that's a huge advantage. If you switch to a different virus, then you have to do more of that testing again.
Got it. I think we may have a question from Kay. Kay, did you want to ask?
Oh, I was just wondering, when I saw that picture of the neurons that weren't joined very well, when you get the therapy, do you have to grow new neurons, or do the old ones get fixed, or what happens?
Yeah, well, that's a very good question, and that's where the in vitro models are not exactly reflecting what happens in vivo, because neurons in culture are per se not as happy as they are in the brain, right? So when neurons get stressed, they start dying, and even the wild type, the healthy neurons, they will die at some point in culture. So neuronal death is not necessarily what you would see at the same time in human patients or with the same pace, right? So for SMA and other neurodegenerative diseases, though, looking at the animal models and also the data from the clinic, what's pretty evident is that the later you treat, the less of an effect you have. So usually, gene therapy, everybody always hopes for a cure, and the picture that we probably paint in our head is people will get up from their wheelchair and start walking. But if the neurons are dead, we can't fix them, right? And there might even also be a point of no return where the neuron is damaged to a point that it cannot be rescued. But what can still happen is that you can either slow down the disease progression very significantly, or you can stop it from further progressing. And so that's kind of how you need to think about gene therapy. It's not if the child had severe degeneration of neurons, we unfortunately at the moment cannot fix that with any technology. Yeah.
Right.
Did that answer your question, or?
Yeah, I just wasn't sure whether you grow new neurons or whether that's it, you've got them and then.
Yeah, no, we can't grow neurons very well yet. Even if you transplant neuronal progenitors, they don't become neurons because the most of the development of the nervous system happens in utero, so prenatally. And then when the child is born, most of the growth of the brain and spinal cord is mainly the maturation. So the cell connectivity, the cells grow in size, and then the glia cells, so the astrocytes are growing. But a lot of the neurons are already there. There are very limited areas in the brain, as far as we know today, that are still making new neurons. And so if you bring in neuronal progenitors, you might get a little bit of a beneficial effect, but they will not become neurons in the patient or in a mouse model because they don't have those developmental clues that you need, during development to know where they need to grow to. That's the problem.
So the best somebody like my child could hope for is just a stopping of the seizures, but no sort of cognitive gains. She's 10 years old.
Mm-hmm. So, well, for this, I think, for the seizure disorders, it is interesting because the question is, are the cells dying or are they not functioning? And so if they're just not functioning properly, but they're still there, then there is a chance that there could be improvement, cognitive improvement. My expectation is that, if we're fixing the problems and the cells are less stressed and they start to function better, a key aspect of the treatment would be to help the child to learn. It will probably almost have to go through some of the learning phases, that the child wasn't able to participate in properly. And so it will probably take some time, is what I would say. But if the cells are alive and we can make them more healthy, then there is a chance that there will be a cognitive improvement, I think.
Oh, that's given me some hope. Thank you.
Yeah.
Thank you.
Kathrin, are you aware, you kind of touched on a little bit, whether neurons are dying versus, not working. I think the data isn't conclusive out there, but I think SCN2A is a little bit unique where the neurons kind of seem like they're surviving, just not functioning right. Is that a fair statement?
Yes, I mean, that's, as far as I can tell, that's the current, opinion, that it wouldn't be degenerative or not as degenerative as other disorders. But, that could also be mutation and specific, some mutations that could have a more severe impact than others. And one aspect that sometimes leads to neuronal death is actually the inflammation. So seizures, continuous seizures cause inflammation in the brain. And when you get those microglia real excited, that can be problematic. That could be a reason why sodium phenylbutyrate could be interesting, because it does affect inflammation as well.
I think that's probably a great transition into some of your mitochondrial stories. And you mentioned that there's higher respiration, which is kind of makes sense from a seizure standpoint. The cells are, kind of going through a lot. Is that, is that higher respiration a kind of seizure related phenomenon, or is it directly due to the mutation? Is it kind of a little bit of a chicken or the egg there?
Yeah, it's a good question. So we actually see it in multiple seizure disorders. So I do think it's not only related to SCN2A mutations. And there are publications out there that look at mitochondrial abnormalities in autism and seizure disorders. And I think I've seen numbers as high as 30% of the patients with autism or seizure disorders would have abnormal mitochondrial function. So I think it might be a consequence, right? So if the ion uptake in the cells is disbalanced, that will affect the mitochondria, because the mitochondria are also involved in storing calcium or releasing, ions into the cells. So work very close with other compartments in the cell. But the good part for therapies is, and I think that's where a lot of the MDs struggle, and maybe also a lot of the basic scientists struggle. It doesn't necessarily matter for a therapy to work whether it's a cause or consequence, right? It's a phenotype. The cells, have a problem with this. So if we fix this problem, the cells might do better, and that could be, that could have a beneficial outcome. A lot of basic scientists and MDs, they're like, but we don't understand why, Actually, we just submitted an NIH grant, and that was one of the main comments we got back, because we didn't get the money. It's like, we don't understand why, we see it's working, but we don't understand why. I'm like, well, that's what we're trying to find out. And, when you look at therapy development, even in spinal muscular atrophy, which is a very simple disease, technically, loss of function, all the patients, have a similar type of disease progression, not all the patients, but most of the patients. we still don't really understand what the protein is exactly doing. It has multiple functions, some are better characterized, but we still don't know why the motor neurons are dying and other cells are fine. And that didn't halt the development of multiple therapies. So I just think, sometimes people are too focused on trying to know everything. Every drug has multiple targets. It doesn't just do what you think it does. So
very, very insightful, and I couldn't agree more. I think Jenny is raising her hand. Go ahead, Jenny. Maybe. Jenny, do you still have a question?
Yes, I'm sorry.
No worries.
Sorry, the phone rang. Yeah, I was just curious with the copper ATSM trial in the pigs. If we really get to start it in the fourth quarter, how long after that, assuming that the data is good, how long is the process after that with the FDA and everything to possibly start clinical trials?
Yeah, so they were asking for a 3-month study. So once we get the animals, we'll have them for 3 months. We have to process all the tissues, of course. So that takes time. Pathologist needs to review all the tissues. That's probably another 3 months, I would say. And then we have to compile all the documents. And, here it's a little bit more complicated because there is a company involved. So the drug that we're using is owned by a company. So it also depends on how quickly they are moving. And, they'll have to review all the documents before we can move into a clinical trial. And that's kind of a little bit of a black box for me, because each company functions a little bit different. Some of them are very fast, very efficient, and some of them are, unfortunately, they might have other, priorities, or they don't have the staffing to go through all of this. I think if we see that it is promising, we don't see any toxicity, then it will be very crucial also to have families of SCN2A work together, to really promote their interest. And then the second aspect is the finances for the clinical trial. So coming up with the finances, is the company willing to pay for a clinical trial, or do we have to find money externally? So all of that can, and it's very hard to give you a timeline. I mean, best case scenario, if everybody pulls on the same string, I'd say probably by summer next year. But if we're struggling with, contracts and if you have to go and try and find money, then it can be much longer. So
Right. No, I get that. It would be nice if we ever get to have an in-person conference again. We still have a contract with the hotel in Columbus, so
yeah,
it would be great timing if we could make that all work together, wouldn't it?
Absolutely. No, I would love to host you all in Columbus. It would be great. Yeah.
That would be fun. Well, thank you. I know a lot of it's out of your hands, so
yeah.
Hope for the best.
But what I'm hoping for is to really if we can find some of the mechanism, then maybe we can develop something as well on that end.
Yeah, that's great. Thank you.
Carla, did you have a question?
I do, and I think you touched upon it with answering Kay's question, but can you give an example, or maybe some examples that we might hope to see patients improve with the compound that you're testing? Or is that too hard?
I think what we need to do is more mouse studies. This study was still very small because also Jennifer is working really hard on testing additional potential therapeutics, which is great. So that kind of limited what she could do. So we're going to continue this in my lab now. And so what we'll have to see is it just survival, or do they have less seizures? Are they improving in their cognitive outcomes? We don't know yet. what I would say, what I would hope so is, that it could have a cognitive impact, because the astrocytes are very important to support the neurons in their function. And if they don't function properly, then the neurons are not going to be able to do their job. So, but we do see copper 87 works on both the neurons and the astrocytes. So it's really hard to say what to expect. I think we just need more studies. I think it's too risky to try and make a prediction. Yeah, I'm sorry.
Oh, no, it's fine. You're correct.
Yeah.
And then I thought of a question when you were just speaking earlier, when you were saying if families show interest. So what are examples of families showing interest to show that our community is interested in moving forward? Is it something like as simple as participating in the foundations like CTRS, because you'll have all that data? Do you know examples?
So yeah, I think what's really important is to raise awareness, but then also work on the natural history, try to gather as much information about the patients that are out there. And then also, one thing, of course, that makes diseases always more interesting for companies, and I hate to say that, is the frequency, how many patients are there that we could potentially treat. And that is something that we don't exactly understand for SCN2A. So I think, additional work that can go into, networking, connection, worldwide establishment of those numbers would be something that could be helpful to get more and more companies interested in SCN2A. And then, just having a strong advocacy group, I think, towards the FDA, but also towards the companies, is always helpful. Like, don't let them get away with saying no. I mean, it's, but it needs to be obviously in a good way, create those connections. I mean, most of the people I work with in industry, they are genuinely really good people. They want to help. It's just sometimes that they have to consider the interest of the company as well. They can't just go and do whatever they want, and then the company goes bankrupt because they spend all the money, right? So that doesn't work either. But I think it's helpful to connect with the companies, make sure they know about the disease, follow up, follow through. That will be super helpful. And then, of course, if participation in research, I know it might be a little bit frustrating for some families because it doesn't lead to a direct benefit necessarily, but it will help the researchers to understand the disease better. And, with that, we will be able to hopefully develop additional strategies to move forward.
Thank you.
Hey, Kathrin, you mentioned kind of participating in, and you guys are doing your iPSC cell lines. Sometimes the families are, asked for kind of samples from a bunch of different labs. If we get like one iPS cell line made for our child at one lab, are those samples kind of like universally accessible between labs, or is it important that, we give samples to like all the labs that ask? Like, yeah. Can you comment on that at all?
Yeah, so unfortunately, there's not a single answer to it. So we have very good experience in collaborating with other institutions, and we have very bad experience where it's basically impossible to get material transfer between institutions. So I think it's important as a foundation, but also as a family, to talk to the researchers that you are planning to give the cells to, and make sure that they are willing to share. And maybe, when you sign that consent form, that it's already written in there that the samples can be shared with other researchers. Because sometimes, even if the researchers want to share, it's the institution that says, wait a minute, in your consent, that's not written. We have to reconsent people, and then you lose easily half a year with that. There are also options, to donate the cells to biobanks such as Coriell or other biobanks, and from there we can buy them. So technically it's a little bit more complicated, but then it will be universally accessible for anyone, even internationally in research. But I would say, if you connect the researchers ahead of time, they can work with each other. The only thing I would say is a lot of researchers, they get the iPSCs, which are the stem cells. But in our case, we're using a different reprogramming method. So I'd say make sure that the skin cells from which the iPSCs are made are also banked, because they are very valuable. They haven't been changed, so that's the primary source, and people can use that in multiple ways.
Got it. Yeah, that makes sense. One follow-up question. Some of our variants, our kiddos variants are, they're repeating. If a lab has, another kiddo's cells of that variant, is it important for us to submit our kids that may have the same variant? Is there a lot of genotype phenotype correlations where it's still important that, there are levels of redundancy in there, or kind of a larger N value, or maybe any comment on that?
Yeah. So actually, yes, because there could be what we call disease modifiers. So in particularly, what will be interesting for us, if there is a patient that has a certain mutation and has a severe phenotype, but then there is a patient that has the same mutation and that has much milder disease progression, we can use this to try and find modifiers again that can lead us to drug targets. And for example, in ALS, that happened that the very same mutation in one mouse model that had a certain genetic background was very severe, and in another model was mild. And they were able to find some druggable targets by just comparing the difference between the genetic layout between the mice. So definitely, just because you have the same mutation, as you all know, I'm sure, doesn't mean that the disease progression or the presentation is the same. One thing that's really important is to, have a good description and a good documentation of the disease in the patient, so that we can link that potentially to some of the observations we make in the cells. And that is a drawback if you work with a biobank, because they will only have information at the time you submit the samples. They don't do follow-ups, right? How did the patient progress? So that is the most interesting part for us as well. How is this patient developing over time? And you're not going to get that through a biobank.
Yeah. Checking to see if there are any other questions. I feel like I'm hogging the Kathrin time here, but okay, go ahead.
Just real quick, Kathrin, you know that Simon's Searchlight has collected quite a few blood samples from our community and created iPSCs. Mm-hmm. Would you be able to access those iPSCs and use in your research, or would you prefer to have the skin? Because theirs came from blood.
Yeah. So the blood cell lineage is very different from the skin and neuronal lineage. That is one thing I'm a little bit worried about. Having said that, we haven't tested, blood-derived iPSCs, but we couldn't do the direct conversion with this because we only get the end product, right? We get these pluripotent stem cell clones. So while they can be valuable, I would definitely prefer the skin cells. I know it's a little bit more of a procedure. It's still on our plan to do a side-by-side comparison and develop a direct conversion protocol from blood cells, because I know it's easier in some cases to convince patients to contribute blood than skin. But we don't have the data. I can't tell you if you would be able to see the same thing. Now, for the seahorse analysis, for the energy metabolism, you might be able to see it in the blood, in fact. So you could just basically look at the monocytes directly. But that would be, you would need the primary material, or the PBMCs, like the peripheral blood monocytes or other cells of the bloodstream.
Okay, thank you.
So the skin, maybe I can just add, the skin sample is very small. It's just 3 millimeters times 3 millimeters. I'm European, I don't do inches, but it's tiny, it would be just like the size of the tip of your little fingernail or something. And I mean, you wouldn't necessarily, you wouldn't have a visible scar or anything that would be, you know, problematic or something like that. So just, it's very tiny. So
it's, our kids have dealt with far worse than that. That's nothing for our kids.
yeah, yeah.
And
they have I'm sorry.
Oh, I'm sorry. I found that the skin punch was actually a lot easier than the blood draw for my son when we did it for a previous study. So I prefer the skin punch. It doesn't seem as scary as people describe it.
Yeah. No. And they have an ectopical cream that they put on, which is, you know, inhibits pain. So it should really be not too bad, I think, in most cases.
Kathrin, when we think about SCN2A, I mean, at least I think about it kind of almost as like 3 different diseases. You have loss of function, gain of function, and then somewhere in the middle, or you're not sure if they're really quite gain of function or loss of function, kind of a mixed function. You know, for some drugs that requires, you know, differing drug profile that could treat it. From a gene therapy, you kind of can think about it a different way. Are there ways or strategies that a gene therapy could be kind of a one drug treats all? Like, could you have a gene replacement with like a microRNA embedded in there that you could knock down that one and replace it? Or
yeah.
Are there anything in the pipeline that could maybe be beneficial for all SCN2A kids?
So, you know, reduce and replace is what you say, basically. It can be done if the gene fits into the vector, right? So you can basically reduce the endogenous levels, whether they're healthy or not. It would be, usually we say it's ubiquitous, but let's say if you happen to have a mutation in that microRNA binding site, your child might still not respond. But the problem with SCN2A is, to start with, that the protein is too big, right? So we can't bring in the entire protein. And that limits that kind of approach. So now you could do, if you look at, for example, the RNA splicing, you can take pieces, right? Depending how big the piece is, you can combine that with a small hairpin. So you can reduce toxic protein that is made and just replace it. But at the same time, if you reduce the RNA levels, then you'll have less that you can fix. So it wouldn't be as easy. The only way that a one size fits all could work is if you could just basically replace the entire gene on the genomic level, right? Because then it will cover all the mutations and you could just swap it out. We're not able to do that right now, but that would be, you know, if you could do that, that would fix for all the patients. But I think for now, you'd probably look at either multiple approaches, at least gain versus loss of function. A loss of function could work for upregulation of the second copy. That could work for pretty much all the patients that have a loss of function. And then reduce the function of the toxic protein could work pretty much for all the gain of function, but then they'll still have the problem of not having enough. But it would be a milder phenotype, most likely.
I think Kay has a question. What if you don't have the gene, such as like a deletion? Would a replacement strategy be best for those type of kiddos?
Yeah, so if you have a large deletion, most of the times that's, loss of function. So replacing will be usually the best case, although sometimes if it's a partial deletion, and you could still make a small protein. And because this is the channel, you know, you could actually have that residual protein piece be included in the membrane and do something wrong. So in some cases, even a deletion can be a gain of function mutation.
Kathrin, you know, Duchenne muscular dystrophy is, you know, been turned into a gene therapy. The dystrophin gene is the biggest gene in the whole genome. For them, they were able to find like a small truncated version that works kind of like that. Has any work been done for SCN2A to look for maybe truncated versions that still have, you know, viable activity?
So, yeah, we thought about it. and not only we, I think, you know, families of SCN2A has an incredible network of researchers. The problem is, so the dystrophin gene is very big, but the middle of the gene acts like a spring. So it's a shock absorber for the muscles. And so that domain in the middle is very repetitive. And, you know, you can imagine if you have a very long spring, it might work better. But if you shorten it, it still has a bit of that same function, right? In the case of SCN2A, you are embedded in the membrane as a channel. So the function of each of the different sides of the protein are very well defined and are unfortunately quite critical, you know, for the function. So if you can't just remove a piece and still make that nice hole in the membrane and still be selective for the sodium ion. So that is the problem. It's a quite sophisticated protein that we're dealing with that is in a very specific location. So everything needs to fold in exactly the perfect way to create that channel. So I think it's very complicated. What could be tried is to say, okay, let's just forget about all of that. Can we find a smaller protein from algae or something that has a similar function just to basically fix, let's say if it's a loss of function, can we just find a protein that can do the same but is smaller, right? But the problem is that this channel is heavily regulated, right? So it has both in the inside the cell and outside the cells, there are binding partners that will regulate how this protein works and when the protein will be active and when not. So unfortunately, it's quite complicated, I think. I think it will be very challenging to find a smaller protein to do the job. I'm not saying it's not possible. Maybe somebody will find it, but that person would be really amazing, you know.
I think Kay might have one more question. Kay or Carla?
Oh no, I didn't. I think somebody asked the question about the deletion. My daughter has 3 sodium channel genes deleted. So okay. What you were saying earlier was interesting, you know, about, but you know, we still can't put the whole gene in. So that was, yeah. One thing you said was interesting because I have a friend whose child has a deletion like my daughter, but inside her deletion, he suffers a lot more than she does with seizures. And when you said maybe a part of the gene is left, I'm thinking he might have a gain of function instead of a loss of function because of that extra piece of gene. I think Sienna's is complete deletion, where I think he's a bit of one of the SCN or SCN2A genes left. So that might be why he presents worse than she does. So that was interesting. Thank you.
Yeah, correct. I mean, I think it's, you know, the cells are quite amazing what they do sometimes with these types of mutations. And it's not, oftentimes it's not what is expected. So, and I can tell you one example for Duchenne muscular dystrophy that we developed. So my husband actually developed a clinical gene therapy for this based on the observation of a patient. So, so Duchenne muscular dystrophy can be very severe or some patients are very mild. And in the clinic, they did find a patient that had an extremely mild phenotype, was still ambulatory at the age of, I think, 60 or 70 years. But when they looked at the mutation, he had a stop codon very early in the gene. And they're like, how is that possible? You know, this person makes protein. You shouldn't make protein. So they started looking for what's happening. And in fact, while the mutation was there on the working copy, there was a mechanism that allowed the machinery that makes protein to just enter at a later time point. And he made probably a protein that's almost 90% full length. It just misses that first piece that carried the mutation. And based on that strategy, they came up with a splicing correction that they were able to use in the clinic. And the clinical trial started, I think, last year or a year and a half ago. So it might help patients that have mutations in that first part of the gene. You know, it doesn't have to be the same mutation. So these kind of things are very interesting. So we always, when we get cell lines, we don't only look at the genetic code, we also then look at the RNA levels. So are they making a different RNA. You know, sometimes the cells, the signaling of that working copy is changed, and then they do something crazy with it, and suddenly you have a smaller toxic RNA that does something it shouldn't do, or you still make protein, it's just truncated. So all this stuff, you know, you can't predict accurately just by looking at the mutation, unless if the entire gene is gone, then most, you know, it's not there, then that is pretty telling. You know, the cells can't do something with the gene if it's gone. Yeah, but if it's partial, there could be all kinds of things happening.
Wow. Thank you.
Kathrin, with the gene therapies, it seems like there are kind of a few different routes of administration. You know, you could do it intrathecally, you could do it systemically, like Zolgensma. Some companies are, you know, delivering it directly into like the deep brain regions. How do you think about that for SCN2A, where, you know, there's clear, you know, it clearly has to be delivered to the brain, but, you know, some of our kiddos also have like kidney issues and GI issues. Like, is it important that it goes everywhere? And I guess what's the best, where the most data reside for those? Because Zolgensma was approved to give everywhere, and I don't think it's been approved to give intrathecally. Are there issues with that type of delivery, or any thoughts?
Yeah. So the intrathecal studies are still ongoing. Actually, the FDA has released the, so there is a study with Zolgensma intrathecally in 3 different doses. And the FDA had put the clinical trial on hold for the highest dose only. Not because of what has been seen in the clinic to that day, but because of some preclinical data and new findings from other groups that showed that high doses of vector in the spinal fluid can actually affect the dorsal root ganglia. However, you know, many studies have shown that this is mainly a problem that we see in the non-human primates, because usually we give them much higher doses compared to the number of cells that we will give the patients, because we want to create that safety margin. And none of the patients that has been treated intrathecally, at least in the trials at Nationwide, as far as I know, or in the intrathecal Zolgensma trial, has had any symptoms that would indicate there is a problem with VRG toxicity. I personally love the intrathecal delivery, because based on our study, you can use less virus and get the same spread throughout the nervous system. In the intravenous delivery, you know, for SMA is only approved for infants under 2 years of age. And the reason is because when you dose into the bloodstream, you have to consider the weight of the patient. So it's dosed per kilogram body weight, and because the blood goes everywhere, right? And there is a threshold above which that is not tolerated because of liver toxicity. All the blood obviously flows through the liver, and the liver is very porous, and it actually takes up a lot of the viral dose. And so that's why you can't, well, you can extrapolate it to older patients, but not at the same dose level. Let's say if in the kids you do 10 viral particles per ml per kilogram, you know, in an older patients, you would have to go down to 2 viral particles per ml per kilogram, just because the overall vector that you give is going to be much larger. And if you do that, you're not going to get enough in the brain. That's the problem, through the bloodstream, right? The CSF delivery, though, allows us to deliver. We have those patients up to 10 years at Nationwide with intrathecal delivery, and that has been well tolerated. The intraparenchymal, so directly into the brain, I'm less of a fan of that, because I don't like, personally for me, and maybe because I'm not a neurosurgeon, but making holes into the brain of kids, it sounds very scary to me. But the problem also is that you don't get the same diffusion. If you do that, you're depositing the virus directly into the tissue, not into the fluid that surrounds the brain and basically spreads it. So then you would have to make multiple injection sites to be able to target bigger brain regions. The intrathecal delivery, actually, according to our data from non-human primates, will still get some of the virus into the periphery. So you target muscle, you target intestine, you'll get some into the kidney. The question is how much. But I think, for me, that will probably be the most likely way to go, will be intrathecal. And I agree with you again, people are just a little bit narrow-minded because they only look at the neurons and say, oh, we need to express this in excitatory neurons in the brain, end of story. I don't believe that's the case. We also have RNA sequencing data that indicates that SCN2A is expressed in the eye. It's probably expressed in some other organs as well. And so you will get a better effect as long as you don't have toxicity in cells that you don't want to target. You'll have a better effect if you target more cells and if you spread it more broadly, in my opinion. Yeah.
Makes sense.
But the GI issues could also be secondary to the neurons that connect to the gut not properly working, right? So that could be, it might not be because you need expression in the gut. It might just be you need to fix the neurons that innervate that area.
Makes sense. What else do I got? Your trans-splicing, I've never heard of that before. That's very interesting. You mentioned kind of it seems like it works, but the biggest bottleneck is either kind of driving up efficiency. I guess how attainable is that, and are there other people working on it? Maybe just any more you can provide on that technology.
Yeah. I mean, it's a very elegant solution, right? That's why I love it. And I'm an RNA biologist by training, but I haven't seen, even from other groups, I haven't seen publications that go past like 10%. So I think there's just something there that we need to discover to make that work. I don't think I'm at the point where I say we're not going to make it work. It's just, it's pretty stubborn. But before we discovered CRISPR-Cas, we weren't able to so quickly and so easily modify the genome either. The efficiency was much lower, and then suddenly you have a breakthrough and the story is very different. So unfortunately, yeah, for now, I don't think, we have ideas on how we can optimize it, but we haven't had that breakthrough where we would say, okay, now we can really change. It's easier. So one thing that is easier that could help some of the, let me pull up my presentation one more time. One thing that is easier, I'm trying to get to the splicing slides. Well, let me see. I'm just going to quickly pull up the splicing slide instead of having you sit through me going there. All right, put this here. And I'm going to share my screen again. So there's different kinds of splicing modulations, and some are easier to do than others. What we're trying to do here is basically take a relatively big piece, so multiple exons, multiple of these little boxes, and try to replace all of them because we can help more patients like that. So one thing that is more efficient and is already in clinic is called, exon skipping. So if you only want to remove one of these, you don't actually have to provide the entire sequence. You can just basically use small RNAs that block some of the signaling here, and you will remove that one box here. So that's already doable, with antisense oligonucleotides and with gene therapy. You can also, include small exons. So all of that can be done. The bigger issue is bigger pieces that would help more patients at a time, or pieces in the beginning or the end of your RNA sequence. So those are the bigger issues. So if you have a mutation here, or here, that is problematic, more problematic to play with compared to removing a piece here or switching out a piece in the middle.
Makes sense. For kiddos that have gain of function, you know, there's antisense. I think there probably is the most data for drug delivery to the brain, but there's also kind of RNAi technologies out there now. Intellia is delivering CRISPRs to kind of go in and permanently break one gene. What do you see as kind of the most promising out of kind of the newer technologies to kind of break a gene or downregulate, and what should be on kind of a parent's radar to be looking for?
Well, it's a tough question. I mean, if you think about durability and long-term treatment, I always like treatments that are more permanent or easier to take. So the problem with the antisense oligonucleotides is if you have to do repeated intrathecal injections, you know, over multiple years, I think that is just something that is not very convenient. I mean, if it's what you have to do and it works, that's great, but there can be some complications with that as well. That's why I like the viral approaches better. So the hairpin RNAs, that will be more permanent. The drawback there is, again, you know, there could be unexpected toxicity that we don't pick up in a non-human primate model. That's related just to the RNA sequence. So they're a little bit more risky at times because there could be toxicity that is hard to predict. But yeah, if I had a choice, I would go for more permanent, just so you don't have to be in the hospital continuously, and you reduce that risk. You know, it's a one-time injection, and then, you know, you don't have to keep tapping the CSF. I mean, compared to an oral drug is a very different story. You know, if there was a drug that can, you know, do the same orally, that's not a big deal. You know, that would be something that would be advantageous for sure, or, you know, anything orally or ectopically or whatever. Yeah, I think, you know, it depends. So for each of these therapies, you would have to evaluate how efficacious is the treatment, because each of these antisense oligos or shRNAs, they're not always equally efficient. So there could be an antisense oligonucleotide that allows to skip, let's say, or to reduce the protein levels by 20%, versus an shRNA that reduces the protein levels by 70%, then I would want to go with the shRNA. But if the oligo reduces the levels by 70% as well, then that might change your decision. So I think it really depends on the type of data that the different companies have. Now, one thing you'd have to be careful is that, you know, especially with the gene therapies, they might exclude you from some of the other clinical trials. So if you do an AAV gene therapy, then maybe the company that develops the antisense oligo doesn't want to include your child because the data will be very difficult to interpret, you know, at least in the initial clinical trials. So that can be a little bit of an issue.
That was going to be my question.
Yeah. Yeah.
Are there any things that would preclude you from doing other things?
So for, you know, if it's an ASO treatment, if you look at Zolgensma and nusinersen or Spinraza, which is the ASO, now that they're both approved, I think a lot of the patients take both or decide to, you know, try combination. But yeah, in the beginning, in the first phase 1, if you are on another genetic therapy, that might exclude you from another clinical trial. It's not that big of a deal with small molecules, I don't think, or any, like, if you just try some supplements or things, you know, those can be washed out. You just say, okay, we're going to stop taking it for a certain amount of time. But the genetic treatments, even the antisense nucleotides, you know, that's several months that they will last. So that would be more complicated, I think.
I have one more on ASO and RNAi, and I'll relinquish all my questions. The ASO seems like they're able to kind of get into brain cells pretty easily, that they don't need any kind of targeting system. The RNAis that have been approved, I think mainly directed to the liver, and have kind of like a GalNAc conjugate on there that allow them to get in there.
Mm-hmm.
Is there any kind of GalNAc for the brain that allow that we kind of for the future that allow those new RNAis to get into brain tissue?
Yeah, I mean, there's maybe not GalNAc, but so the non-viral gene therapy field, which that goes into, develops very quickly. So there is a lot of new ideas on how you can coat those particles. So basically you put your RNAs or your DNAs in lipids or protein molecules, and they can be, they can have certain tags that would make them more specific for certain cell types. And there's a lot of research that is going on that. And then the second part is, one thing is the coating of those particles, but the other thing is how can you make the spread through the spinal cord more efficiently? And that's something that people are working on with different methods. You can tilt the patient head down at the time of injection, try get that spread going. There are different formulations of your solution, more viscous, less viscous, with sugar in it, with no sugar in it, and all kinds of stuff. But there's also, for example, potentially development of devices that will help with a pump, for example, to get things moving faster. So there's a lot of, I think, innovation around that area that can still happen and will probably have a major impact on the efficacy of these treatments moving forward. But to the best of my knowledge, from what I've seen, viruses are still more efficient in getting into the cells than non-viral gene therapies. But that might be just a transient thing, with all the research that's going in non-viral gene therapy, that could change at some point. Yeah.
Thank you, Kathrin. We're kind of getting close on time. Are there any important topics that kind of didn't come up that you think are important for the families to know, Kathrin?
Any other things? Well, I think, so one of the things that you heard a lot about it is the gut-brain axis. So I think that's something to really pay attention to. I think I strongly believe that modulating the health of the gut could have an impact in seizure disorders, just because there is so much inflammation that can originate through the gut. And there's a lot of signaling that goes on between the gut and the brain. It's not going to potentially cure a child from having a disease that impacts the neurons, but it could really, in terms of having an effect on the symptoms, some of the symptoms that I think are difficult to manage. I think we need to put more energy in taking this more serious. I don't know what your experience is, but I feel like sometimes in the clinic, this aspect isn't taken very serious yet. And I feel like this will be something that could really support the health of the kids.
I don't know if I can speak for everybody, but I think I probably can. I think this is a constant topic among SCN2A kiddos is gut problems. And when gut problems get bad, then seizures get bad. There always seems to be a really tight correlation there that we have a hard time. We end up going to a GI doctor, and then they try to fix the GI problems, and the GI doctors never really talk with the neurologist, and we never really get very far. So a really interesting topic.
So I think that is something that really needs to improve, the multidisciplinary teams in the clinics. I think that would be really something amazing. And I know Nationwide is putting a lot of efforts that there is communication between the different departments, but I think that is an area definitely to improve. The other thing that I find really puzzling and don't know necessarily what to do with is that I've heard from a lot of families that the seizures are basically linked to certain times of the day or certain situations that are repeatedly the same. So either in the morning or in the evening when they need to fall asleep. And I feel like we need to find ways to better analyze what changes in the cells during that time of the day that causes those seizures or allows the seizures to occur. And if we understand that, then we can potentially also develop therapeutic targets for that.
Fantastic. Thank you, Kathrin. Really appreciate everything today. Super exciting, super interesting. And you're just so knowledgeable on so many different topics. A really fantastic interview and talk. I know that Leo has some words to kind of close out the talk, but I just want to thank you personally.
Thank you.
Awesome. It was great. As always, I love talking to Kathrin. All right. So as always, that was an amazing session. Thank you all so much for joining, and Kathrin for volunteering your time to help us understand your work a little bit better. I mean, it just gives us so much hope for our kids' future. And thank you to all the families that joined today and asked questions. They were very insightful. And if you're like me, you're going to go home and you're going to think about this. You might watch the recording a couple times. And as you digest the science, you might come up with some more questions. So we're always here for you, the volunteers and the board members and the staff at Families SCN2A. Just send us an email, call us up, and we'll do our best to get those questions answered for you. There are 4 more table talks in this RX Hope virtual series, and each of them is set up just like this. So you're welcome to ask questions, and it's open discussion. So be sure to register for those in advance. We're going to make these recordings available as soon as possible, and Carla, when she makes them available, she always puts some links at the end of the recording for you to click on and check out some other things. And, you know, if you heard what I heard, it's just so clear that we have still so much work to do, and we can't do it without fundraising and your participation in research. So one major way to get involved right now, if you haven't done so already, register for the SCN2A clinical trial readiness study. It's our job to be ready for when these things come to clinic. Just go to scn2a.study and sign up today. And I think that's it. So I hope you have a great rest of your week and weekend. Thanks again, Sean, for moderating.
Thank you, everybody, and have a nice day.